Hi there, my name is Brian Minalga, presenting on behalf of the Legacy Project at the Office of HIV/AIDS Network Coordination based at Fred Hutch in Seattle. And this is our poster, the Representative Studies Rubric, 12 Steps to Address Underrepresentation in HIV Clinical Research. We know that women and other underrepresented populations suffer the consequences of exclusion from HIV research. When we talk about underrepresented populations, we're not only speaking of cisgender and transgender women, but also other transgender persons, persons who inject drugs, pregnant persons, as well as Black, Indigenous, and Latinx people in the United States and territories. And when we speak of consequences, we can point to specific examples of cases where populations excluded from clinical research have not had the benefit to have a product or drug approved for their population due to their exclusion from the clinical research of that product. This has happened in recent years with PrEP and other HIV treatment and prevention modalities. So in order to address this issue of representative study populations, our team designed a 12-item questionnaire that can assess individual study protocols for their inclusiveness in terms of age, ethnicity, gender, drug use, pregnancy, race, and sex assigned at birth. For example, question 2 on this 12-item questionnaire asks, are cisgender women eligible to participate in the trial, yes or no? If the answer is no, the questionnaire asks, is there a reason or justification stated for the exclusion of cisgender women? So there are 12 such questions on the RSR, or Representative Studies rubric. After we designed this questionnaire, we applied it to 47 HIV clinical research protocols that were enrolling across the NIH-funded HIV/AIDS clinical trials networks. These are the ACTG, HPTN, HVTN, and IMPACT networks. We selected these 47 protocols because they represented 100% of protocols that were enrolling at the time of this analysis, which was September of 2021. When we applied these 12 questions to these 47 HIV clinical research protocols, we found that a significant number of these 47 studies excluded women and gender minorities with no justification for exclusion provided. I'll direct your attention to the red bars on the chart on the poster. These represent studies where the population in question was excluded with no justification provided in the protocol. So for example, there was one study that excluded cisgender women with no justification, 18 studies that excluded transgender women, 25 that excluded trans men, and 32 that excluded gender non-binary people with no justification provided for the exclusion of these populations. So clearly gender minorities face a high rate of exclusion with no justification provided within the protocol. We also found that the vast majority of studies incorrectly defined the study population by gender identity and sex assigned at birth, and that over half of the studies required participants to actively avoid becoming pregnant, which results in the exclusion of people with pregnancy potential and the continued unknown of how study products work and or are safe and effective in people with the potential to become pregnant. In conclusion, we found that this tool, the RSR, the Representative Studies rubric, is a simple screening tool that helps to identify and rectify components of HIV research protocols that unnecessarily exclude women and other underrepresented populations. We presented our findings to the leadership of the 4 NIH-funded HIV/AIDS clinical trials networks, and the leadership endorsed our recommendation to implement this screening tool, these 12 questions, proactively as a protocol development tool to ensure that our clinical trials are designed to enroll representative study populations. To learn more about the RSR, including access to the full 12-item questionnaire, scan the QR code on the poster and get in touch with us. Thank you so much.